In part 1, we tried to answer the where part of the question about this coronavirus. We got up to speed till December 2019 and went through the evidence to examine where the virus could have come from. Now, we will try to answer the what question. What virus was this? There are two main theories about the origin of this virus. One, that it jumped naturally from wildlife to people, that is, a naturally emergent virus. The other, that the virus was under study and experimentation in a lab. There is an abundance of information and evidence for this that I have come across, but I want to share with you four of the most important pieces in my opinion. An absence of diversity. Let me explain. When viruses circulate through a given species, they keep changing through mutation and recombination. Recombination is when two viruses attack the same cell and then recombine within. This can be seen now amongst us humans. We are seeing multiple variants of the virus due to mutation and recombination. This happens quite quickly too. We started seeing variants of SARS-2 within 3 months of the first outbreak. So if another species, say pigeons, had to now get this virus from us, there would be multiple pigeons getting infected from multiple humans and there would be multiple variants. But when SARS-2 broke out, there was zero evidence of multiple hosts infecting multiple humans. All initial cases had the exact same genome structure with zero variation. The evidence points to a single point of outbreak, a single variant, a single event, a single patient zero. The natural evolution of viruses also poses a significant question here. It's the spike protein in coronaviruses that determines which animal cells they can attach to and attack. Viruses don't just jump from species to species. The spike protein has to evolve step by step through many mutations for the virus to become a dangerous pathogen. For example, in SARS-1, after the virus had gotten from bats to civets, it took 6 changes in the spike protein before it could mildly affect people, 14 more changes to fully adapt to humans, and 4 more to become an epidemic. But in SARS-2, the virus has hardly changed at all. From its very first appearance, it was well adapted to human cells, well adapted to infect and to spread. I have provided links to a couple of science papers which describe this in greater detail. Furin cleavage. This is probably the most important clue to the nature of the SARS-2 coronavirus's origins. The coronavirus spike protein has two subunits, S1 and S2. The S1 unit latches on to the cell and then the S2 penetrates the cell structure. But for this to happen, the S1 and the S2 have to separate. They have to break off. Human cells have a protein cutting tool called Furin on the surface. It will cut any protein chain that carries the right sequence of amino acids. So if you put something in between the S1 and S2 that the furin itself can cut, then that aids the virus in doing its job perfectly each time. This is called a furin cleavage site. Virologists have known for 20 years that the quickest way to enhance a virus is to give it a furin cleavage site between S1 and S2. There are at least 11 gainoffunction experiments in the public domain, including by Dr. Shi Zheng Li, which focus on adding a furin cleavage site to a virus to enhance its potency. One of the sequences that furin recognizes is proline arginine arginine alanine, or PRRA as it's denoted using alphabets. If furin sees PRRA, it will cut that protein chain exactly at that point. SARS-2 is the only coronavirus which has this cleavage site. No other SARS-related beta coronavirus has this site. There seems to be a 4-letter insert right between the S1 and S2 protein chains. Those 4 letters in SARS-2 are PRRA, perfectly tuned for furin's preference. In April 2012, 6 miners got sick while working in the Mojiang caves in Yunnan province. They developed COVID-like symptoms: coughs, fevers, headache, chest pains, breathing difficulties, and 3 eventually died. Dr. Shi Zhengli's team then isolated a SARS coronavirus called RATG13 from rats in those caves. She released a paper about this virus and said that this is the closest known relative of the SARS-2 virus. The paper was released only in February 2020, a few weeks after the Wuhan outbreak, even though they had the virus for 7 or 8 years. I downloaded the full genome of RATG13 and of SARS-2. It's just a string of letters. This data is available in the NCBI database. I have provided a link for this in the description. There are 1,269 letters in the genome of the spike protein of RATG13. There are 1,273 letters in the genome of spike protein of SARS-2, which means it has extra 4 letters. There is indeed one insert of 4 letters in the SARS-2 virus. Those 4 letters are PRRA at the Furin cleavage site. So let me summarize this. They have had this RATG13 virus for over 8 years now. They have done gainoffunction experimentation on it. And it was common knowledge that a Furin cleavage insert would be the fastest way to make a virus more potent. And then there is a Furin cleavage insert in SARS-2, making it the only known beta coronavirus to ever have one. And that insert is PRRA, for the fusion to identify and cleave perfectly. How many coincidences would it take to shake your belief in coincidences? There are many more points and arguments which I have provided links to in the description, but these ones stood out to me and I have shared them here with you today. After nearly a year and a half of not finding any evidence to support the natural emergence theory, the lab leak hypothesis has gained massive momentum over the last 2-3 months. Whichever way you lean, you would agree with me that there is enough evidence for a deep and thorough investigation into all possible theories. The good news is that it seems to be headed that way, with more and more people and countries demanding an open and detailed investigation. The records of the Wuhan Institute of Virology certainly hold much relevant information. WHO has stated that they will do a full investigation, and there is mounting pressure on Chinese authorities to provide access to the data and records of the Wuhan Institute. Regardless of that, the truth is that there are viruses in captivity that could debilitate the world. We have seen firsthand what could happen if one virus emerges or if one is leaked. And we need to ensure that this doesn't ever happen again. Milton Lightenberg's article in the Bulletin and more recently Nicholas Wade's article in the Medium, some great pieces of scientific journalism which actually got me started on this research. In maybe about 200 hours of research, I've had the chance to see such brilliant work by so many individuals and organizations. For example, there is an ingenious statistical analysis by Dr. Stephen Quay, who looks at data in a very unique perspective through patient admissions. His analysis shows how all the initial admissions of COVID patient clusters were in hospitals along the Wuhan number 2 subway line. That subway line runs from the Wuhan Institute of Virology, passes the seafood market, passes the high-speed train station, and finally ends at the international airport. The high-speed train station connects Wuhan to other parts of China, and the international airport has daily flights to major large cities across the globe. His work shows a realistic model for the spread of the virus through the city, the country, and onward to the world. Fascinating work on data and interpretation. I highly recommend it. Each source, document, science paper that I have referred to is linked in the description, and I encourage you to go down these fascinating rabbit holes. Before I end this video, I want to leave you with some food for thought. In the comments of my last video, someone pointed out that in the history of pandemics, over the last 100 years, the country of origin has always been in the top 10 in terms of cases or fatalities. I went back and checked up on that. After all, China has the largest population in the world too. So one would imagine that China would be in the top 20, top 30, top 50. As of today, China is in the 97th position in countries listed. 97th. About 91,000 cases and around 4,600 deaths. Two things are possible here. Either those numbers are untrue, which would then thicken the cloud of doubt over any info that we get from them on this entire pandemic, or those numbers are true, which is scarier. Did they do something different that no one else in the world did? Or did they know something different that no one else in the world did? Your guesses or theories are as good as mine. We live in a dangerous world. Take good care of yourself and those around you. I'll see you next time.